Okay, I think we should get started. It's 1 after the 9, so Sandy told me we can begin. I brought notes because our speaker is from the UK and they have very elaborate titles in the UK, and so I need to make sure that I read this correctly. It's my pleasure to introduce today's seminar speaker, Professor Adam Sharp. Adam is the Wellcome Trust Clinical Research Career Development Fellow and lead of the Translational Therapeutics team at the Institute for Cancer Research in London. He's also an honorary consultant in medical oncology in the prostate cancer targeted therapy group and drug development unit at the Royal Marsden Hospital. That's why I needed my note. That's pretty something else. So Adam initially trained as a biochemist and a pharmacologist at the University of Southampton. So he obtained his PhD degree there. And then he subsequently went on to do medical training there. So it was not a combined MD PhD program, it was a PhD followed by MD training. After that, he became a clinical research training fellow at the Medical Research Council, and he worked under the supervision of Professor Johan de Bono, who's well known to many of us. And Adam was prolific in Dr. de Bono's lab from 2015 onwards. And he's now an independent investigator. He has significant publications and funding in various aspects of prostate cancer research, AR signaling, mRNA translation, apoptosis signaling, which is what he's going to be telling us about today. He's won numerous awards, research awards from the Royal Society of Medicine, the Academy of Medical Sciences, and the American Society for Clinical Oncology, or ASCO. He's also been awarded a Prostate Cancer Foundation Young Investigator Award. That's how I got to know Adam as a friend and colleague, and he's gone on to be PI of Challenge Awards as well. And we're very fortunate that he was doing a secondment or a sabbatical of sorts for the summer in the lab of Steve Balk at Harvard University. And so he's been in the US and was kind enough to join us here at the University of Minnesota before he heads back, and that will be later this week. You'll go home to your colleagues and your clinic and your family and your friends. And so thank you very much for making this pit stop to share your work with us.
Well, thank you very much for the introduction, Scott, and thank you for the invite. And it's a real pleasure to be here and see all of you guys in Minnesota and tell you some things really that we've been doing over the past, I think, 5 years on apoptosis. And every time people read out about it, it just makes me feel older. And yeah, anyway, so as Scott said, so I run a lab. So it's a wet lab, it's a mechanistic lab, and we're really focused on identifying new targets or repositioning drugs that have made a difference in other cancers into prostate cancer. And then clinically, I do only clinical trials. So I do phase 1 to phase 3 clinical trials. So I think I'm very fortunate in that space. I don't do any standard of care. I just do clinical trials. And today I want to tell you about some of the work we've done over the last 5 years that's really been looking at targeting the intrinsic apoptosis pathway in lethal prostate cancer. So these are my disclosures, none of which relate to today's talk. And for many of you, I don't need to tell you about this. I think really this is just to say that prostate cancer is a common disease. And I think for me also is to just really focus on that red bit. I've realized being in the US that many of my colleagues actually work on the spectrum of disease from early disease to late disease, but both myself and Johan, Professor de Bono, we really work on late stage disease. So the men that we see have had all lines of therapy. They don't really have any treatments left at all that have any efficacy. And these are the men that we see in clinic. And I think I just wanted to highlight that. So these are the guys on the right-hand side. They have a huge number of metastases. They have very few treatments. And we're really trying to understand the biology of that disease to develop new treatments to make a difference for these men. And that's kind of what we've been doing over the past 5 to 10 years. And I think just for an introduction is obviously the management of advanced prostate cancer. This is a simplified slide, but I think it just shows that over the last 10 to 20 years, we've made a real difference. We've developed a huge number of therapies that have efficacy for these patients. But I think the challenge is this disease still remains fatal. And I think for me, this is where we can really make a difference, is by understanding the biology to develop new treatments for these men with advanced prostate cancer. And this is also really just to give you an idea of where we work. So just really going back to that contextualization about the landscape of disease. So if you think on the right-hand side of this slide, this is seeing patients after they've had all of these treatments and they have very little treatments left. So these are men that have the PSA increasing, radiological progression. And then if you can see, we're very fortunate. So we really do bed to bedside, I think, and kind of lab to bedside in terms of our research. So the cancer biomarkers team is Johann's team. So they're very focused on developing new treatments, biomarker assays, reverse and forward translation. We have my team, which is the translational therapeutics team. So this is really looking at the kind of mechanistic biology of prostate cancer and trying to develop new treatments. And then we're very fortunate because we actually have an academic drug discovery unit. And I think that's really quite cool. So we actually have a team at the Institute of Cancer Research, about 120 people that really do drug discovery and drug development. So we can take our ideas, our targets, and trying to develop drugs to them. And then again, I think I'm fortunate as an oncologist because I also work in the drug development unit and the prostate team, which means that we can take those ideas to the clinic and try and make a difference to men with advanced prostate cancer. And I think, as Scott has alluded to, I'm extremely fortunate that I have a huge number of collaborators around the whole of the US that have really supported me. And I think I've been very fortunate to kind of stand on the shoulders of many, many giants. So how do we make a difference to these men? And I think this is a slide that some of you that have seen my talks have seen before, is so how do we address this? And I think for me, it's critical to understand the underpinning biology. I don't think we do this enough. So what is it that drives the prostate cancer, drives progression and drives resistance? And identify those patients where the biology happens. You know, everyone will suggest, well, this is going to cure everybody with prostate cancer. It very rarely is. So where does this happen? It's great, you've got some cool biology, but which are the patients that biology happens in? And then develop, this is my kind of plea, my soapbox, develop therapeutic approaches based on biology, not convenience. There are so many trials that happen because people just have those drugs and it's not really based on the underpinning biology. And then change that discovered biology from a negative to a predictive biomarker. There is so many studies that will tell people they're going to do badly. You know, myself, other clinicians, you can tell patients that are going to do badly. And what you want to be able to do is say, you've got this, it means that we think you're going to do badly, but can you transform their disease by giving them a treatment based on that? You know, let's think about HER2 in breast cancer. It used to be a bad prognostic biomarker. It's now something that is positive because there's many drugs targeting it. And I think we need to do that more. And then the hardest thing is to deliver those biology-driven hypothesis testing trials. It's not enough just to say this is important. Let's actually deliver it to the patients and try and make a difference to them. And clearly that is the hardest bit. It needs more funding and many of those things fail, but it's the only way we're going to impact our patients. And what I hope by doing this is we can discover and develop novel treatments for prostate cancer patients. So this is kind of a summary of what I've just said. So prostate cancer is common, you know that. People in this room and around the world have changed the treatment for prostate cancer. You know, androgen receptor pathway inhibitors, taxane chemotherapy, now radionuclides, PARP inhibitors and immunotherapy. We've developed treatments that improve the survival of men with advanced prostate cancer, but it still remains fatal. And we need new efficacious treatments. And I think that's an urgent unmet clinical need. And again, on the right-hand side, this is where we work. So these are patients that are progressing. They've been through all of their treatments and they have no efficacious treatments left. And I think for me is can we understand the biology of that disease to deliver biology-driven approaches? And I think for me, that will change those from being really incremental to potentially transformational. And that gets me onto what I want to talk about today. So can we harness the intrinsic apoptosis pathway? So apoptosis, programmed cell death, can we kill the prostate cancer cell by modulating the apoptotic pathway and deliver prostate cancer specific cell death? And that's really what over the last 5 years, one of the 3 pillars of my lab, which Scott alluded to. So we still work on AR signaling. Disclosure, I still think it's the most important target in the prostate cancer cell. Apoptosis and RNA translation. And today we'll focus on the apoptosis work that we've done. So a quick summary, intrinsic apoptosis, it's programmed cell death. It's regulation of the cell death in the cell. And we have the BH3 only proteins and they're pro-apoptotic, so they drive apoptosis. And that's countered by the BCL2 family. So MCL1, BCL2, and BCLXL. And what they do is if you think you've got BACs and BACs that are on the mitochondrial membrane and they're pore-forming proteins, and they're what allow cytochrome C to be released and drive apoptosis. And what we've really been interested in the last 5 years is can we identify strategies that can drive that prostate cancer cell to undergo apoptosis? And ways that we can do that is we can inhibit the BCL2 family itself, or could we activate and sensitize the cancer to BH3 only proteins? And can we tip the balance towards apoptosis and drive prostate cancer cell death? And I think, apoptosis is not, it's not a new concept. It's been around for many, many years. And I think we know in prostate cancer that actually as patients progress to castration-resistant prostate cancer, to treatment resistance, we do see increase in these anti-apoptotic proteins, MCL1, BCL-XL, BCL2. And if you look on the bottom left-hand side, that's a lymph node of a patient with castration-resistant disease. They're full of anti-apoptotic proteins. That's MCL1 by immunohistochemistry. And these allow the prostate cancer to survive. You've got inhibition of those pore-forming proteins back and back, and they support cell survival. Now, the other side of this coin, as well as a clinician who's looking to translate things to the clinic, was that it's a real hotbed of drug development at the moment. So historically, BH3 mimetics have been developed. So for example, BCL2, venetoclax, which is approved for hematological diseases. And now there's BH3 mimetics targeting BCL-XL and MCL1. And I think more interestingly, because there has been some toxicity problems with those drugs, is there's now antibody drug conjugates and PROTACs coming to these targets, which can actually try and help alleviate some of the toxicity and take these strategies as we develop them to the clinic. And I think for me, one of the most interesting things is that if you inhibit all three of these, if you look at the bottom right-hand side, these are patient-derived organoids. And these are the patients that I've just talked about, that I've told you about. These are patients that we see in our clinic that have had multiple lines of treatment, that are resistant to everything. And we take a biopsy from them and we develop organoids. And what you can see is if you inhibit the anti-apoptotic proteins, you invariably get induction of caspase 3-7 and you get cell death. So these cells can still undergo apoptosis. We just need to work out how we induce it. And the challenge, of course, is to inhibit all of those three proteins is going to be associated with toxicity, because normal tissue is also reliant on these proteins. So what we've really been working on over these last 5 years, and I'll talk to you about today, is trying to really deliver specificity and try and really deliver prostate cancer specific cell death through novel strategies. So the two stories that I want to give you a little bit of insight into today is one that has been published, which is the one on the left-hand side. So that's looking at prostate cancers that are really defined by BCL2 family proteins, and that will look at BCL2 positive lethal prostate cancer and its role in lineage plasticity. And then on the other side of the slide is something that's unpublished, but will hopefully be published very soon, which is where we've really looked at MCL1, which is another one of the BCL2 family proteins, to try and identify cancers that may be vulnerable to single agent MCL1 inhibition or synergistic combinations with other approved therapies. And I'll go through both of those today and give you a little bit of background to what we've done. So the first one is a little bit shorter because it's already published and you can read it for yourself. It was published in JCI at the end of last year. And what we identified is that BCL2 expression is enhanced in advanced prostate cancer with lineage plasticity. And we really got onto this because we had a real in-depth look at androgen receptor immunohistochemistry in prostate cancer patients. And what we showed that if you take men that have castration-sensitive disease who develop castration-resistance disease, and this is paired samples, so this is from diagnosis to castration resistance, is as you would expect, you get increase in AR full length because you know that most of them get amplifications, they have other aberrations within AR. But what we really wanted to look at is this, how many of them actually lose AR? Because you'll see now when people talk about prostate cancer, they'll give you the landscape of disease and they'll say that everyone dies with neuroendocrine. That is not the case, I think, and we really wanted to look at AR as a hard marker of who really loses AR and who really does badly. And what we've shown again is it doesn't actually happen that often. So on the middle, you can see 187 metastatic biopsies. And what you see is that actually only less than 5% actually lose expression of the androgen receptor. And as you would probably expect, is one of the main treatments being AR targeting in our patients is if you lose the androgen receptor, you do badly. And that's not unique, but it was probably the biggest cohort of patients that people had really looked at immunohistochemistry for the androgen receptor. But the question for us was, it is a small population, but they do badly. So how could we potentially target that or target that particular subset of cancers? And Professor Nelson at Seattle had previously looked at BCL2 in patients that had neuroendocrine disease. So we also did the same, and we were doing it kind of alongside his work. And what we showed is that patients that lose AR, that are AR negative, have much enrichment for BCL2, one of those anti-apoptotic family proteins. And then orthogonal data looking at RNA-seq from the Stand Up to Cancer PCF cohort, as expected, showed that BCL2 associated with markers of kind of neuroendocrine or lineage plasticity, EMT, JAK-STAT signaling, but also you saw downregulation and de-enrichment of the androgen receptor. And then unsurprisingly, is those cancers that had increased BCL2 expression did worse. And now those of you that have been observant is you're probably saying, well, that's because it enriches for AR negative disease, which we know. But interestingly, when you actually go and look at them and try and break them down, is whether you have AR positive or AR negative disease in the context of BCL2, you seem to do worse. So those that have BCL2 high and AR negative and those that do BCL2 high and AR positive respond less to androgen receptor targeting therapies. They have less time on treatment and they have a shorter overall survival. It's very small numbers, but it starts to make you think, does BCL2 just provide a marker of those cancers that have started to undergo lineage plasticity towards a more basal neuroendocrine subtype? And if you think contextually about the prostate, is actually BCL2 is extremely highly expressed in basal cells. So it might be that these are just reprogramming their transcriptional program back towards a basal lineage plasticity phenotype. And on the left-hand side is just an interesting observation. And I think, again, stuff that we would know, but it's nice to see. This was a biopsy from a patient that we took. They had actually a dual pathology in their biopsy. They had neuroendocrine and adenocarcinoma. And this is just cutesy, pretty, I quite like it, is, so the AR full length, you can see you have huge expression in the adenocarcinoma, nothing in the neuroendocrine. BCL2 is flipped the other way around. So the AR positive adenocarcinoma has no BCL2, the AR negative neuroendocrine has high BCL2, and the Ki67 matches that. The neuroendocrine is higher proliferation, the adenocarcinoma is lower proliferation. And what was really nice is actually when we developed the PDX, it had clonality at the start. It had both of those clones. And as you would expect, as time went on, as you castrated the PDX, the neuroendocrine disease grew out, and that's all we were left with. And again, just to really mimic what probably is happening in a patient. And then we did some work with Dr. Hafner at the Fred Hutch, and we just really looked to see what may be regulating this. And he's done a lot of in-depth stuff looking at methylation. And it seems that in those neuroendocrine cancers, you see hypomethylation of the BCL2 promoter, suggesting that maybe it becomes more active. And then again, with his support, we looked at ASCL1, which is a common transcription factor involved in neuroendocrine differentiation. And we saw in those neuroendocrine cancers that what you saw is much more ASCL1 binding at the BCL2 promoter. Again, just giving hints at how maybe this pathway is regulated. And then, of course, the big question for us is, well, how could we potentially translate this to the clinic? So we took all of our cell lines, and you can see them there, and we took the H660s, which are probably the only bona fide neuroendocrine prostate cancer cell line, or very difficult to work with, which have high levels of ACL1, high levels of BCL2. And we showed that if you knock down ACL1, you get down regulation of BCL2. Again, just kind of reinforcing this potential mechanism of regulation. And then when you treat those prostate cancer cells with venetoclax, which is an FDA-approved compound, you see caspase induction in those neuroendocrine cells, and you see a huge shift in the cell viability in those neuroendocrine cells, again, suggesting that maybe venetoclax is something that we could explore in neuroendocrine prostate cancer. And I think that's really where we are now in terms of this approach, is really thinking about a kind of proof of concept, proof of mechanism study, whereby we could maybe explore venetoclax in neuroendocrine disease. The challenges of that is, of course, it's a relatively uncommon population. We've really focused here on AR negative, which probably makes it even more uncommon. And it's getting buy-in from companies that want to do that in such a small population, but I think it's definitely worth exploring. And that's published in JCI. So if you want to have a look at that in more detail, then please feel free. And if you have any questions, please reach out to me. So for the main bit of the talk and the last kind of 25 minutes is I really want to talk about some new work that's not published yet. And this is focused on, so what we've shown you is that a very small population of prostate cancers overexpress BCL2. Actually, adenocarcinoma has no BCL2 at all. The main drivers of adenocarcinoma are really MCL1 and BCLXL, so the other two anti-apoptotic proteins. And we'd kind of focused on MCL1 for a reason that I'll come on to in a minute, is that it's actually amplified in a lot of prostate cancers. And what we wanted to really understand is, based on what I told you, is the challenges with toxicity, is can we identify genomic backgrounds that sensitize to single agent MCL1? Or can we explore combinations whereby we target tumorigenic pathways and actually sensitize with synergy to MCL1 inhibition? And I'll talk through two of those strategies that we've really worked on over the last 4 to 5 years. So I think the challenge with MCL1 inhibition itself is, and I've already alluded to this, is if you think about the 3 anti-apoptotic proteins, we can kind of ignore BCL2 in an adenocarcinoma context because it's rarely expressed. But if you knock out MCL1, you still have BCLXL. So you have redundancy between these two proteins. So there is functional redundancy, and that can be a challenge in terms of developing and delivering a therapeutic strategy. So what we did is we looked at our cell lines and we looked at single agent MCL1 inhibition. And again, as me and Justin were talking about this morning, is we're very poor in prostate cancer and we don't have many cell lines. So, these were the 6 that we looked at. And what we saw is you take 2 different MCL1 inhibitors, AZ5991 and S63845, and you saw that actually LNCAP95 and 22RV1s are actually quite sensitive to single agent MCL1 inhibition, induction of caspase 3,7 and cell death. So it seems that those cell lines do have the ability to undergo apoptosis in response to MCL1 inhibition. And we then did a bit of a deeper dive, not too much, but we looked at, well, what's the MCL1 status of these cell lines by FISH, and we showed that 22RV1s have copy number gain, and this is associated with increased protein expression. Interestingly, DU145s also have copy number gain, and an interesting observation is that they actually don't undergo apoptosis at all, even with the positive control inhibiting all of them. And I think that's because they've actually lost a pore-forming protein. And I think when you start to work in a space and you deep down into mechanisms of cell lines, you realize that a lot of these cell lines have things that you never realize. So actually DU145s themselves can't undergo apoptosis. And that's because they've lost one of those pore-forming proteins backs. And then LNCAP95 didn't have MCL1 amplification. So the challenge with biomarkers always is they're impure, and there's never a perfect biomarker. So 22RV1s may have an enhanced response because they have MCL1 amplification and they have a reliance on it. But LNCAP95s actually are active, they respond, but we don't know why. And we still don't know why today, and that's part of ongoing work within the lab. And also, without giving, VCAPs are the most exquisitely sensitive. They just look at an MCL1 inhibitor and they die. And that's something that me and Steve Bulk are also very interested in following up. So having looked at our cell lines, seen that there may be some enrichment for MCL1 amplification, we went back and looked at our samples. And this, I think, is an interesting both observation and finding. So what you can see here is really looking across 3 different cohorts. So the TCGA, those of you working prostate and other cancers, what a resource that has been. The Stand Up to Cancer, again, for those of you working in prostate cancer, what a resource has that been? So that's primary prostate cancers that, in TCGA that really have been cured, or the majority have been cured. Stand Up to Cancer is those metastatic guys like the ones that I treat that sadly will die from their disease. And then Joaquin Mateo, who worked with Johan and led many of the PARP studies, also did a paired cohort. And I think that's really important. Comparing TCGA and Stand Up to Cancer tells us very little. They're just born differently. But looking at those patients that we have primary disease from that developed CRPC, they're a very different population. And as you'd expect, AR is kind of my sense check. I always like a sense check because I'm not an informatician, as I was talking to Justin earlier. And as you would expect with AR, there is very little AR amplification in primary prostate cancer. Metastatic disease, clearly focal amplification. And in the paired cohort, it's the same because we induce AR amplification by our treatments. But we wanted to look at MCL1. So MCL1's interesting is, in TCGA, it's 3% amplification across the arm. And in Stand Up to Cancer, it's about 15%. Now, what I will say is this is actually a chromosome 1q amplification. It's not a focal amplification. It's part of chromosome 1q is amplified. But interestingly, in the paired samples, so those samples that we had from patients that they were born to get CRPC, the amplification is the same. So those guys, it seems to be an early event in men that get bad disease. And that's just highlighted here. So if you take the men that we see, so this is retrospective, they've got CRPC, so we know they're going to get CRPC, and you go back and get their primary tissue, it was there at the start. And I'm not someone who treats early disease. I'm not someone that works in that space, but actually could this be a biomarker for intensification? So if someone comes that's got high risk primary disease and you identify an MCL1 amplification, they're pretty much born to go and get CRPCs. And again, I'm not suggesting an MCL1 inhibitor, but could we be thinking about other intensification with other treatments? And I think this just brings that out really with support from friends. So if you take the TCGA cohort, and this is all the publicly available data, and you pull out those cancers that have MCL1 gain or amplification, you don't really see this they do really badly. They fall off. They're the guys that get progressive disease. They're the guys that aren't cured. And again, if you go and look at markers of bad disease, the size of their tumor, the number of lymph nodes, the Gleason score, they all had bad disease. So again, it seems to highlight those guys that have bad disease. And then Dr. Zawalski, Adam Zawalski at the NCI helped us. We just looked back in his kind of neoadjuvant cohort. So these are high risk patients that he gave intense neoadjuvant treatment. And what we saw is really that, again, those ones on the right-hand side that had MCL1 gain or amplification really didn't get complete responses. So again, it just suggests that maybe MCL1 could be a biomarker of those that do poorly at diagnosis. And then in metastatic disease, and I always say this, is again, in the stand up to cancer, it does seem to predict those people that do worse. But my caution always with stand up to cancer or our own cohorts is these are bad. You're looking for bad, good versus bad, bad. Sadly, all of these men will do relatively badly, and biomarker studies can be challenging. So seeing any kind of difference is interesting, but there's lots of heterogeneity. There's lots of, there's lots of confounding factors, but it's interesting. So we kind of seen in our cell lines that MCL1 amplification may enrich for single agent MCL1 treatment. We'd seen clinically that it may be relevant. I'm sorry, I always use may. I don't like to overcall things. So it may be relevant. And then we wanted to go back to our patient-derived xenograft and organoids because, again, we've got really poor numbers of cell lines to really try and understand if that amplification did enrich for response. The pathway on the left is kind of our part. So, take the biopsy into a mouse, and we do it that way. Johan many years ago did lots of primary organoids from metastatic disease, and it was just challenging. It's easier from a PDX because there's more tissue. And then we looked for MCL1 amplification, and we also looked for immunohistochemistry, and we then did the same experiments. And this is really just to show those who are clinically minded and also scientifically minded that these are 5 patients that had aggressive disease, had numbers of treatments, and we developed PDXs from all of them. And what we did is we went back and retrospectively looked at which ones had amplification and which ones didn't. And we found 2 that had amplification and 3 of them that didn't. And then we performed the same experiment. So you can see on the right-hand side is this fish to show whether or not there was copy number gain amplification. And those 2 that did have increased amount of protein by IHC. And they seem to have a higher induction of caspase 3-7 in response to single agent activity. And they had increased cell death. So again, just suggested in another small number of cells, this is now only 5, but more than the ones we had before. So we've now got 10 that single agent MCL1 may be enriched for response if you've got MCL1 amplification. So that's kind of the first part. So you can now consider, well, is the rationale for patients that turn up and they have primary disease and they have MCL1 amplification, could we think about intensification of treatment? And in those that have metastatic disease as MCL1 inhibitors hopefully become available, although there has been some toxicity with the BH3 mimetics, but they are developing ADCs, could that be a therapeutic approach? I think for me, it's always, could you increase the population? Because that's only 16% of patients. And what we wanted to do was understand if we could use combinations to still deliver cancer-specific kill, but maybe broaden out the population that we could treat. So what we did is we took the MCL1 inhibitor and we went and did a combination screen. So what we wanted to do was look at those combinations that potentially could activate or enhance MCL1 activity. And we took an FDA-approved screen. You could go and do a CRISPR screen, but again, I am a physician scientist, and it's very difficult to develop 2 drugs that are really not approved. So we did an FDA screen because we wanted to look for approved drugs that we could take to the clinic with an MCL1 inhibitor. And what we showed was that PI3 kinase inhibitors really synergize with MCL1 inhibition, increase caspase 3-7, decrease cell viability, and we didn't see that in normal cells. And at the time, the likes of Johan, Manuel, others were developing AKT in prostate cancer. We weren't really going down the PI3 kinase route. So we switched to AKT inhibition. And that clearly at the time wasn't in the FDA approved screen because it wasn't approved. And we showed that, as expected, that the synergy with AKT inhibition. Actually more synergy, but again, what do you call more synergy? It's kind of zip scores, there's synergy. I think that's the best we can say. And then outside of the screen, because you always have to validate what you've found, is we validated what we saw. So on the left-hand side is LNCAP95s, on the right-hand side is C42s, and these are exactly laid out the same. So what you can see is that if you treat LNCAP95s with vehicle, CAPI, or IPA, so 2 AKT inhibitors, you get very small induction of caspase 3-7, actually none at all. AZD 5991 is one of those MCL1 inhibitors. You'll see in LNCAP95s, there is slight induction because I showed you they're already sensitive to single agent to a degree, but we don't see that in C42s because they're not. But when you treat with both, there's huge induction of caspase 3-7. And then that's mirrored in the cell viability. You can see a huge increase in cell death with the combination. And then my lab always get frustrated with the number of orthogonal validations that I want. So then they had to do Western blots and show me that you're getting beautiful induction of cleaved PARP, cleaved caspase 3, and showing that we really are driving apoptosis with this combination. And then at the bottom is IHC. We don't routinely do IHC to confirm our findings, but we wanted to validate the cleaved caspase 3 assay for in vivo data that will come later. And you can see exactly the same on the right-hand side with C42s. And for the rest of the talk, just for time's sake, is I'm going to focus on the C42s, but we've done it in multiple cell lines. So then the question is okay, so you've shown that MCL1 and AKT inhibition synergize, but actually how's that happening? You know, so mechanistically, what's happening? So we already know that MCL1 inhibition inhibits MCL1. And I've told you that in adenocarcinomas, we don't really see BCL2. So BCL-XL became our focus. And the presumption and the hypothesis was that something we're doing with AKT must be blocking BCL-XL. And again, you could go and do a genome-wide screen and try, but actually I think we wanted to be, you know, hypothesis driven. So why not look at the things that modulate BCL-XL first? Because, you know, if you hedge your bets, you would hope that that's what's happening. Now, we know that BAX and BAC, of course, the pore-forming proteins. So if you're affecting those, that could affect apoptosis. But also the BH3 only, so these things that are pro-apoptotic, we presume that we could also be affecting those. So actually, we just did a small siRNA screen on the most likely things to be impacting this pathway. And what we showed is, so we did the same experiments, but with knockdown of the individual members of the pathway. And again, just to orientate yourself, this is caspase 3-7 assay. You've got DMSO, you've got AZ, which is the MCL1 inhibitor. You've got the 2 AKT inhibitors, then you've got the combinations with both. And the white bars are those, you know, that's the effect of the combination on its own. So the induction of caspase 3-7 up to around 15 to 20-fold. And what we saw is that if you knock down BIM, BAD, and BAC, you actually get rescue back to levels of AKT. And remember, AKT, contrary to popular belief, doesn't really induce apoptosis. It causes cell cycle arrest. So you still get the effect of AKT. And you rescue back to the effects of AKT alone. And what was really nice is then when you do cell viability alongside it, you also rescue the cell viability effect back to AKT alone, suggesting to us that, you know, BIM, BAD, and BAC were the key players in this mechanism that we'd discovered. And that makes some sense. You know, MCL1 is known to be a more dominant regulator of BAC, and you know that BIM and BAD are involved in regulating BCL-XL. But again, we need a little bit more of a deeper dive to work out what was happening. But the first thing we wanted to do was take all three of those and really show that we could completely rescue the effect. And that was what was really nice. So if you, again, this is C42s, just repeating the same data, is you look at on the left-hand side is caspase 3-7, and we've knocked out BAC, BAD, and BIM. And again, with vehicle, you see no induction of caspase 3-7. With the combination, you get really nice induction. And then in both combinations, you knock out BAC, BAD, and BIM, and it completely rescues back to baseline. And the same with cell viability. But again, with cell viability, you're seeing a little bit of effect, and that's just the AKT inhibitor on its own. And then on the right-hand side, because of my, you know, slightly obsessiveness, the Western blots is again really nice. You see that cleave PARP going right up, that cleave caspase 3 going right up. But if you knock out the 3, it completely rescues the effect. So the cell death has completely disappeared. So then the question is, well, we kind of know where our targets are and where the money is, but how is AKT affecting those? Because we think that's what's happening. And that's what we did on the right-hand side. So you now take all of those experimental conditions, and what you see is that there's no real effect on BAC. There's no real effect on total BAD. There's a little bit of effect on BIM, but not huge because it's really highly expressed. But what you saw is that you're affecting phospho-BAD. And again, that's known. You know, AKT is regulating phospho-BAD. So in the AKT inhibitors or the combinations, you get downregulation of phospho-BAD. And this is actually another aside, and I don't want to waste too much time because I've got lots to get through. But that's interesting, right? We think about AKT in prostate cancer. We very rarely look at functional biomarkers. So could phospho-BAD be a good biomarker of PI3 kinase AKT activated cancers? Rather than genomic changes, which could be, I don't know, not functional. And then the final thing with the mechanism is, of course, the dynamic. So we now BAD, BIM, and BAC. We believe that that's really where the money is. So then we did lots of IPs and lots of interaction studies. And what we showed is, the kind of hypothesis was that we're going to block BIM MCL1 with the MCL1 inhibitor. when bad is phosphorylated, it sits in the cytoplasm binding 1433, which is a protein interaction protein. And when you dephosphorylate, it binds BCL-XL. So we showed that. So when you block MCL1, BIM now, when you pull down MCL1, you get reduction in binding of BIM, as you expect. And when you pull down BIM, you get reduction in binding of MCL1. And what was the key was really what we showed is that when you look at phosphobad, is when you treat with AKT, you get a huge increase in bad binding to BCL-XL. So you're now blocking BCL-XL. So you've now blocked MCL1, you've blocked BCL-XL, you get induction of caspase 3-7, and you drive apoptosis. Now, those of you who've been observant, and at the start I said you can't block MCL1 and BCL-XL because it drives cell death and it's toxic in normal cells and in prostate cancers. I think this is where this got more exciting for me, is actually this is biomarker driven. So when you do this across all of your cell lines, you only see caspase 3-7 induction and cell death in those cancers that have PI3 kinase AKT activation. And what we saw is it's those cell lines that have phosphobad, high levels of bad, BIM, and BAC. And we weren't expecting this. That was serendipitous. But again, it was really nice to see that now this could potentially be biomarker driven. Now, interesting, PC3s are PTEN loss, but they didn't seem to have activation of these biomarkers. And also in other experiments, we've shown that PC3s aren't reliant on BAC to undergo cell death. They're reliant on BACs. And this is interesting. So they don't actually rely on this mechanism to maintain their cell survival. And that was exciting. And then we went back, as we always do now, to our patient-derived models to, again, proof of principle and increase the numbers. So if you take the PDXOs that I showed you, 3 of them are AKT P10 activated. And those are the 4 that respond on the left. So you can see upregulation of phosphobad. If you looked at just pathway analysis of their RNA-seq, they have high levels of pathway activation. And if you want to look at the hard markers, CP50 has an AKT amplification. 253, 267, and 336 are all P10 loss. CP142 is unaltered, but unaltered in terms of the investigations we've done. Of course, there could be something that we haven't seen or haven't picked up. And what you see is those 4 models that have activation of the pathway, they have induction of caspase 3-7, and they have cell death, again, suggesting that this combination will work in a biomarker population. And then hugely grateful to a friend, Marco Bezzi, who works at the Institute of Cancer Research. He's a mouse modeler because, of course, all of our patient-derived organoids have very different genomic backgrounds. They're not homogeneous. And what we did is we took his models, and he kindly showed us that P10 knockout models have high levels, again, of our biomarker phosphobad, and they respond to the combination, whereas those models that don't have activation of the pathway don't respond. And then that's great, because then what we did is we, as the clinician, we went back and looked at all of the cell lines, all of the organoids, all of his models, and we plotted phosphobad versus, the induction of caspase 3-7 and cell viability. And what you get is a beautiful correlation. So those that have high phosphobad respond well to the combination and die, and those that don't. So again, you start to think, could this be a biomarker that we could use in clinic to give patients that combination? And then again, as always, reviewers would want in vivo. So we took one of the models, P10 loss model, CP253C, and we treated it in vivo and showed that the combination was much more effective than vehicle and single agents. So the challenge, though, is how do we translate this? So BH3 mimetics targeting MCL1 have had some challenges, and that's cardiac toxicity, and this is probably on target. So the other thing that is quite commonly out there is that CDK9 inhibition does downregulate MCL1 acutely. So we at the ICR had been involved in developing fadraciclib, which is a CDK9 inhibitor. So again, I'm going to go through this a bit quickly because we're getting towards the end of the talk, but just to really show that we've recapitulated the whole story with a CDK9 inhibitor, because I think it might be a quicker way to translate this to the clinic. And this is really in the same format as what you've seen before. So I'm just really showing the same stuff, but with a different thing. So this is going to be your AKT inhibitors, but with a CDK9 inhibitor rather than an MCL1 inhibitor. You can see in 95 is exactly the same. You get very little caspase 3-7 induction and then huge cell death in the combination. And the Western blots show the same. And you can see the same with C42s. You get a huge induction of caspase 3-7 and the cell viability goes down. And you see the same with the Western blots. And what was nice is exactly so, those rescue experiments, if you knock out all 3 of that BIM, BAD, and BAC, you get rescue of the combination effects. So you get increase in caspase 3/7 without the knockdowns. And then when you knock them down, you get rescue of the caspase 3/7, and the same with cell viability. And again, just to remind you, on the bottom right-hand side is kind of that biomarker panel. Again, the same cell lines seem to have induction of caspase 3/7 and the same decrease in cell viability as we saw with the MCL1 inhibitor. Now, again, I always like to caveat what we do is, of course, we're looking earlier because we're looking at apoptosis. So all of our stuff is done at 24 hours. There is no doubt that that CDK9 inhibitor will start to have effects if you leave it for longer. But again, it does start to think about how you could become dynamic in your dosing within a clinical trial, and you could do things slightly differently rather than having to have these things on all of the time. And then again, going back to the patient xenograft organoids showed the same. So you'll remember I told you that 50, 253, 267, 336 are all PI3 kinase activated. 142 isn't. It's those ones that are activated that have the synergy. So that's the light blue and the dark purple on the right-hand side. Whereas again, the 142 really just seems to have the effects of the CDK9 inhibitor. There's no synergy. And then if you take Marco Betzi's models, which are those mouse models on the right-hand side, you see the same. It's the one that is non-activated is now in red, and you really don't see the same levels of synergy, both at caspase 3/7 induction or in cell death, suggesting the same principle that we saw with the MCL1 inhibitor. And then in vivo, it recapitulated single agent, so the vehicle, again, the black line, single agent CDK9, the blue line, and ipatasertib, the red line, had very little effects compared to the combination, suggesting that maybe in vivo, maybe again, clinically, this would be a good route to translating this study. So in summary, and then a few future perspectives at the end, just to kind of discuss where we're going, because I think it's good for conversations later and questions. So I think for BCL2, I'm quite excited, but I'm also challenged by how we translate it. So for me, from what we've done and from what Professor Nelson has done at Fred Hutch, NAPC would be the group that you would want to target. The challenge with that is, what's your biomarker? I think for me, it would be AR negative and BCL2 high, because that's what we've done. But already you then make that a really small population, because that's not the neuroendocrine differentiated, the lineage plastic. But it may give you the best chance of it working. And that's something that I will continue to talk to. CRPC, adenocarcinoma, I don't think is where I would go with that, is we don't see it expressed. Now, interestingly, Steve Bulk does see upregulation in CSPC, before CRPC developed. So again, could we think about going earlier? But I think we need to do more work to see if that's really true. And is it just transcript going up? Is it associated with protein increase? Because really that's the functional aspect. MCL1 amplification occurs in 16% of MCRPC patients and may identify those with early disease. And I haven't put that we should treat with an MCL1 inhibitor. I'm not saying that, but could we think about whether these are the patients that need some degree of intensification if they are destined to get metastatic castration-resistant prostate cancer? MCL1 amplification enriches for response to MCL1 inhibition in preclinical models of lethal prostate cancer. Could you conceive a way that you give a CDK9 inhibitor or a MCL1 target that we see that could be targeted to those patients? And then MCL1 and AKT inhibition synergizes to trigger apoptosis. It drives cell death in preclinical models of lethal prostate cancer. And we know what the mechanism is, or we believe we do. It's induction of apoptosis mediated through BIM-BAD-BAC. And it's biomarker driven. The cancers have to have PI3 kinase activation. But again, is phospho-BAD a better biomarker, because that's functional and shows that the pathway is activated rather than relying on genomic analysis? Again, more difficult because you'd have to do a phospho assay, which can have liabilities. But I think, again, doing it in a center where it's all validated, I think that has promise. And then does CDK9/2, sorry, the fadraciclib is a CDK9/2 inhibitor, not a pure CDK9 inhibitor, potentially provides an alternative strategy that we could probably get to the clinic quicker, because those drugs have been in pediatric trials and have been tolerated. And then the future perspectives for me, and really where the lab is looking now, is additional biomarkers, additional combinations, and clinical translation, which I think has to remain the focus for me as a physician scientist. So these are just not to give away what we're doing at the moment, but it's always fun and you're all friends. So, additional biomarkers. So left-hand side, interestingly, again, real focus in CDKs. There is toxicity associated with them. They do a lot of things. But I think in prostate cancer, we don't do this enough, is we actually went back and looked at the modulation of CDKs. So and we've done this with splicing as well. So we actually lose, prostate cancer loses a lot of CDKs. So, for example, 10% of prostate cancer patients lose CDK10. So does that then open up a therapeutic vulnerability? And if you take 22 RB1s, and you knock out CDK10 or it's cycling, CCNQ, you get huge sensitivity to MCL1. So again, if cancers have loss of CDK10 in patients, could we treat with an MCL1 inhibitor? Then in the middle is kind of a thing that I think we've lost sight of, is MEK activated prostate cancers. And really for me is, a high level of patients have activation. I think the reason we haven't got too excited about it is because they're not driven by genomic oncogenic drivers, but actually they're driven probably by other things. But what we've shown is, again, that 22 RB1s that are phospho-ERK activated, they have a BRAF mutation. These respond to MCL1 MEK inhibition, whereas cancer cells that don't have activation of the pathway don't. And then finally, in my finish is, we can translate this. That's the CDK9 inhibitor, downregulates MCL1. Docetaxel, a treatment that we use in every day, downregulates MCL1. So there are ways that we can translate this to the clinic and understand it. And then my final point is there are also tumors that are addicted to MCL1. So for example, I've just shown you there is there's about 10% of patients that lose BAC, which is one of the pore-forming proteins. That's associated with decreased MCL1, as you can see here. And they're more sensitive to AKT. So can you use biology that has actually inhibited MCL1 itself, that you can then use the other portion or the combination therapy to deliver activity? And that's kind of some of the ways that we want to go now and try and broaden the potential to deliver these approaches to the clinic. So on time, I just want to thank everyone. So in my lab, and this has really been led by Juan Jimenez Vacas, who's a Spanish postdoc who is fantastic, and Dan Westerby, who's a MD PhD, who's now moved to Newcastle to continue his academic training. They've really led this work. Johan, who continues to be an amazing mentor and has supported me, as Scott was saying, through my postdoc, and still continues to support me, although now, I'm in independence and doing, different spheres of research, including apoptosis. And again, the prostate community, and I don't want to say it in kind of like we're huggy, but we are, I've been so supported, if you think of we are, so Pete and, the guys at the Fred Hutch, the guys at UW, NCI, at Vancouver, Harvard, I've just been so fortunate to be supported by really giants of the field. And then there's our clinical and teams on the right-hand side, those people that have supported me funding it. And to be honest, our patients and families, I've showed you loads of patient-derived models. I've showed you loads of tissue stuff, although the majority of my lab is mechanistic. And without their real support and giving us their tissue, knowing that really they may not get much back is fantastic, and we can do what we do without them. And thank you for the invite, and thank you for listening.
We have microphones for everyone for questions. Here's our first question.
So in first part of your talk, you're showing that the BCL2 expression is high in the NEPC cell line, and you show the ASL1 chip C, they have some binding in the BCL2 promoter. So actually I have two questions. So have you expressed the ASL1 in BCL2 low cell line to see if it can induce the BCL2 expression? And then my second question is, have you looked at the expression level of the BCL2 in NEPC patient compared with the normal CRPC patient to see if the BCL2 expression is super high in the NEPC patient?
Yeah, so two excellent questions. So second one first, and sorry, because I was going so quickly because I wanted to show it, but didn't want to spend too much time on it. We have done that, and that's exactly where I find. So MCL1 and BCLXL are ubiquitously expressed. So that's why in the adenocarcinoma, we very much focused on those. What I like about BCL2 is it really is enriched in, and what I don't mean is enriched in like it goes from high to really high. You don't see it in adenocarcinoma, and you only see it in, and I don't want to say neuroendocrine because we've only looked at AR negative. So I know they are, you know what I mean, so absolutely. So it is very much enriched in that disease. Now, that's why I would be excited to potentially run a BCL2 targeting trial in that subset. But the challenge is that's a very small population. It's bad disease, it's aggressive. But if you're talking to companies and trying to get them involved, it's the challenge of the size of the population. But so that's the first bit. You're right. And I put my hand up is we didn't go into as much detail. And I think for me, I don't, as Scott was saying, I'm an AR adenocarcinoma person. This was data took us into the neuroendocrine space. And I think we wanted to get it out there because what I want to do is try and run a trial. But I do think mechanistically there is more that could be done. And I think you're right, is could you convert an adenocarcinoma phenotype to a neuroendocrine type through BCL2? Now, my one worry is, and I'm always a worrier and a glass half empty, is BCL2 just a marker of lineage plasticity rather than the driver? And I know that the H660s respond well to BCL2 inhibitors, but since then we've also tried some patient-derived organoids, and it's not always on or off. So do you see what I mean? Is if you think about basic pathology, is BCL2 is highly expressed in basal cells, normal basal cells. So I'm wondering, and this is what we talked about in the discussion, could BCL2 just be a marker that those tumors have reverted back to a base? Now, that's good because we don't have good markers of that type of disease, but whether or not it's a passenger and expressed rather than a driver, but your experiments would help support that. Could you change an adenocarcinoma to a neuroendocrine phenotype through overexpression of BCL2? And we haven't done that. Good question.
Thanks. That was really great talk. And I'm sure there's been a lot of attempts to target the apoptotic pathway. I'm going to make an assumption because you didn't talk about it. There's no role for the extrinsic pathway, DR4, DR5 activation. And then the second question is further downstream. Is this story for SMAC mimetics or enhancing, the caspase release in combination with what you've shown?
I think both are very possible. I think, and again, I don't want to be too myopic, but I think we very much focused on the intrinsic apoptosis pathway, per se at the level of the mitochondrial membrane. I think in terms of how we develop now, having built a track record, have reagents in the space, I think both of those are definitely worth pursuing. We've again, it's not that I don't think the extrinsic is important or involved, it's just we haven't played there. But I do think downstream, and whether or not, like you say, is XIAPs and other things could be used and they're coming into the clinic, I agree totally. And I think it's certainly worth following up and investigating. And also maybe more clinically translatable because they may be getting ahead of the direct BCL2 family targeting agents in terms of clinical development. Good question.
Hey, great talk. I just had a question so how specific is all this to really prostate cancer? Like when you introduced all the TCGA data and everything, have you stratified other cancers? Does this fit, or does this really seem like only prostate cancer?
Yeah, Matt, I'm so myopic, and you're absolutely right. And I think I need to get better in terms of grantsmanship and publications. Is absolutely. So there was a paper from AZ, and I don't think they went as in detail as we did. But in breast cancer, for example, in PI3 kinase activated cancers, they showed that MCL1, I think they used mTOR rather than AKT, but again, it's the same pathway. So I think this could have broader implications. And I think you're right, if you were going to run that trial of phospho-bad activated cancers, it would be nice to think about it as an agnostic biomarker from a tumor specific. And you could see, I'm in a drug development unit, we see every single type of cancer is actually, you get their biopsies, you do phospho-bad, and if they're positive, they're eligible for the trial. And maybe you then enrich your expansions in those patients that the biology seems to play. I think that's a great point, and I think we haven't done. In terms of looking at MCL1 amplification, there is data on lung cancer and myeloma already out there suggesting that this also holds true. So I think you're right. Is it something that could become bigger than what I've presented? Prostate's big and it's common, but there are other very common diseases that rely on PTEN AKT signaling. It's good point.
The results showing that BAC-BAD or BIM knockdown rescues that synergy phenotype, that's awesome from a science and mechanism standpoint, but might be bad for a clinical translation standpoint because loss of those factors could end up either driving acquired or intrinsic resistance to your therapeutics. Have you treated any of your responsive models longer term to see if resistance develops and if that resistance is via that mechanism or a related mechanism?
I haven't. I suppose to hypothesize, it would be interesting to see whether or not they become it's not linear, but if you think about it, you've got, where MCL1 back is kind of that side, the BCL-XL, but, do they become more sensitive to the other side of the pathway, and then they now become, a single agent BCL-XL inhibitor. So we haven't, and I think that is, again, something that if we could get traction and we were starting to look at doing, a proof of concept clinical trial, is you'd want to know what's the next step. And I think that would be good in terms of development of this strategy. So what would you then come in and do? So we haven't done. And also the slight challenges, and again, I said it in the paper tongue in cheek, is, this is driving cell death. So there is nothing on the plate. So do you know what I mean? So how, to get them to grow out, you know what I mean? And this is why I like this, is you thinking, you don't give them a chance to develop resistance. Now we know that that's not going to be the case probably in a patient, because there'll be so many different clones and there will be some that grow out. But I think what will probably grow out will be the cancer that doesn't have the phenotype that you're targeting. And experimentally, which is a nice place to be, it's difficult to do the studies because, when we first started this project, we had to do the assays earlier than we wanted to, because we were going to the plate the next day to do the CASP, and there's nothing there. It's really that strong in terms. So good question, but I think experimentally challenging because of the profound effect.
Are there any other questions on the floor?
Yes,
Yep.
great talk, Adam. And it was fun to talk and walk with you. So Adam, it really looks like you're presenting that maybe the patients could be stratified by some measure of apoptotic, or at least priming apoptotic activity. Now, you have really shown nicely that maybe we can think about some phosphorylation markers, etc. How do you think we should re-stratify patients? And are we going to do this through existing tools, or do we have to go deeper and think about phosphoproteomics, etc.?
Yeah, I think that's a great point. I think for this is, I think going back and doing the whole of Atlas, whether it's TCGA or Ellie and Dan's stuff, to re-stratify based on phosphorylation is difficult. But I think within the context, if we're trying to develop and deliver small phase 1, phase 2 clinical trials, I think asking the functional question is not that challenging. A phase 1, phase 2 of that combination would be 30 to 50 patients at most. It's not difficult to get those biopsies and do a validated assay and make sure. I think broader, I think we should be thinking about functionality of pathways. I think how many of these drugs, failed is not the right word, because I think sometimes we fail them. I hear it from Johan, and it's not, oh, we did that, but it was done 10 years ago. There were no biopsies in the trial. And we start to say that strategy is no good because we've run a trial. But did you check what that drug was doing? Was it modulating the pathway that you were interested? Did the patient even have the biomarker? You know what I mean? And I think we as a, you don't want to keep reinventing the wheel. But I think for apoptosis, this is where I also think, you get that criticism, oh, we've done it, but we've not done it in prostate cancer where we have 5 cell lines. You know what I mean is, I think it's much more context specific. And I think we need to know that context. And I think you're right, is those kind of studies where we can answer functionality is going to be really important. But I think we probably have to start in the context of something where we need it. And then maybe with technologies that develop, we can do it in a broader context. Because you would want to, well, how many patients have phosphate? If you're going to start a trial, if I see 100 patients in a year, and I do a biopsy on everyone for phospho-bad, actually you do want to know that landscape stuff, because you're like, how long is it going to take? How many patients am I going to have to see? How many am I going to have to biopsy? So that's where I think the landscape studies will become important, because they will inform on, it's all worthwhile, because the one patient that you find that responds, it's great. But you know what I mean? Is that, yeah, so I think absolutely, I think we do need to go towards more functional assays. And I think within the context of a specific question, maybe to start, but as you guys develop more technologies, maybe we can start to reclassify based on what's functionally important in their cancer.
All right, thank you very much. Let's say thank you one more time to Dr. Turk.